Welcome to Introduction to Ecology, Lesson 20. Up to this point in the semester, we've mainly focused on populations of organisms. Now in the last unit, we did look at pairs of populations, specifically focused on species interactions. But by and large, we've sort of looked at fairly simple systems with one or two species. Now we're going to move on to communities. And community ecology is my favorite because it's messy. It's interacting populations of different species living in the same place at the same time. And typically, because ecological communities are very complex with many different species living in them, community ecology is extremely complex itself. It involves sort of trying to understand a myriad of different interactions among different populations and how that drives their population dynamics and the overall structure of the community. So that's what our focus is on today, community ecology. And to understand community ecology, we first need to know how to define communities. And there's a number of different ways that ecologists tend to define different communities. One way is to base your definition of a community on a structure or some other element of the physical environment. So it could be the group of species that are living on a particular structure. A good example would be the community in a tide pool. So a tide pool is basically just a depression in the rock. It's a physical structure, but it traps organisms in there. And all the organisms that are in there live and interact with each other in the same place at the same time, and that makes a community. And the boundaries of that community are sort of defined by where the tide pool ends, where the water no longer pools inside the rock. Another example might be serpentine plant communities. So this isn't so much a physical structure as a sort of chemical makeup of the environment. So serpentine rock is very unusual chemically, and as a result, different types of plants tend to live on serpentine rocks than live on other types of soil or other types of rock. And so you get this very specialized plant community that then attracts a somewhat specialized invertebrate community, and so on and so on and so on. And so we can talk about serpentine communities because, again, those are defined by where this serpentine rock is found. It's sort of defined by that physical environment. We can also define communities based on a biological element, and in particular, the presence or importance of an abundant species. So we could talk about kelp forest communities, and in that case, the abundant species is the kelp, and that species provides habitat for all of the other species. That's why it's sort of that important ground for the entire community building up is that kelp, and then all the fish that live in the kelp forest and all the invertebrates, the crabs, that sort of stuff that live in the kelp forest, they all interact, but they're sort of there because of the kelp. Another good example that's terrestrial might be a beech forest. So beech forests tend to be very low diversity. Mostly the trees you're going to find are going to be the beech trees. And so these beech trees provide the habitat for all of the animals and insects that live in that forest, both in terms of just the trees themselves, providing nesting sites for birds and food, but also even just the leaf litter on the ground is mostly going to be beech leaves, and so all the litter insects are going to be primarily using the beech litter as where they live. And so again, we can talk about the beech forest communities because, again, we're using those beech as our defining feature of where the community is and where it sort of ends and what exactly the group of species we're looking at are. Now, what you notice from what I've just said is probably that the definition of community, or at least the delimitation of a community, is somewhat arbitrary. I might define a kelp forest community, but I could just as easily define a near-shore community off the Pacific coast, and the boundaries would be slightly different, and I'd be looking at a slightly different set of species, but they'd be overlapping. And so frequently when we define a community, it is a bit arbitrary, and there's some element of art in terms of finding and defining your community to ask a specific question. And so generally we try to pick the community that we focus on based on what we want to ask, what we want to know about that ecological system. One of the reasons that we have trouble defining communities, we need to sort of narrow our boundaries, we can't just look at these huge communities in the entire country, is that communities are complex and they consist of many, many, many species. So even in a system as simple as, say, the community of insects living in downtown Clemson, just making a species list is probably very difficult. There's just a lot of different things living in the same place at the same time. And so it's hard to make a list of everything that's there. You can imagine how difficult it is to monitor them all, to monitor whether their populations are going up or down, to figure out how they're interacting with each other, who's affecting who in what way, which species are interacting positively, which species are interacting negatively, which ones are eating each other. And so that's another problem. And then there's the additional problem that in some of these really complex communities, or even in less complex communities that are understudied, we don't even know what's out there. So we have probably defined about 1.9 million species. But scientists estimate that there's probably about 9 million species currently in the world today. So we don't even know what those are. We've never tried to define them scientifically. And so it becomes very, very hard to study a community where there's things you don't even know exist in the entire planet. So again, that's what makes community ecology interesting, but it's also what makes community ecology difficult. Sometimes we try to subset communities even further, make them more narrowly defined. And there's different ways, again, of doing that. One common way is to subset a community based on evolutionary lineage. So we might only look at the mosquitoes that live in a particular area at a particular time. And the reason we choose mosquitoes is because they're all somewhat related to each other. There's that taxonomic relationship between them. And so that's not always the case, but frequently things that are related have similar lifestyles. Maybe they're competing for the same food sources or the same nesting sites, so you expect some kind of interaction between them. But of course, just because you're related doesn't necessarily mean you interact. And just because you're not related, even more so, definitely doesn't mean you don't interact, doesn't mean you don't compete for the same food or the same nesting sites. And so sometimes rather than defining our communities or subsetting our communities based on evolutionary lineage, sometimes we will subset based on what we call guild. And a guild is a group of species that all use the same resources. They may be taxonomic related or they may not be. So you might imagine all mosquitoes eat detritus, and they're taxonomically related, so you might expect that. But there's a lot of other stuff like snails and other types of insects that also eat detritus and might be competing for that detritus in little pools of water. And so again, that would be a guild where we'd look at all the organisms that are interacting with or using that resource. Here's another example where we have the herbivore community, and herbivores are a feeding guild where something like a fish with a backbone eats the plants, but also something like an isopod, which is basically like an aquatic insect-like thing, it also eats the plants. And so both of these are competing for the same food source, even though one has a backbone and one doesn't. They're clearly very distinct evolutionarily, but they're in the same guild because they're using that same resource. Another way of subsetting is based on functional group, and that's a little bit different than resource or than guild. So a functional group is a group of species that function in similar ways, but they may or may not use resources similarly. And so one example that I've come up with is a community of microbes in an anaerobic digester. So some of these microbes will use different carbon sources as their energy. Some might use glucose, others might use fructose, some might use xylose. They're using different resources to live, but they're all kind of living in the same way. They're all able to live in a low oxygen environment, and so their function is the same. And they might all be sort of contributing to that environment in the same way. They're digesting the stuff that we put into the anaerobic digesters. So again, that's sort of a subset of a community based on function rather than resource use or evolution. All right. So another way that we can subset communities is based on trophic interactions. And we've seen this before. Trophic interactions are any interaction that involves one species feeding on another species. And so we have all of those different levels where we start with primary producers, then primary consumers, secondary consumers, tertiary consumers, and quaternary consumers. Now, often when we subset communities based on trophic interactions, we typically study interactions between those subsets, interactions between our primary consumers and our primary producers, or between our secondary consumers and our primary consumers, that sort of thing. Not always, but that's typically the case. And that ultimately leads to the idea of food webs. So food webs are representations of trophic or energetic connections among species within a community. It's basically a picture of who eats who. You draw lines between the things that are eating each other. So at the lowest level of our food web, we have the primary producers. Those are our autotrophs, or our plants. And then we have our primary consumers, the ones that eat those, herbivores, something like a grasshopper maybe. And then we move up to secondary consumers that feed on the herbivores, that feed on the plants. And then tertiary consumers and all the way up to quaternary consumers. And if we drew lines sort of between those, we'd have a food web. Food webs are great. They show us who eats who, but they're a little bit too simple. They don't really capture the whole messy truth of ecological communities and trophic interactions within these communities. And there's a couple of reasons why they don't. So one thing that food webs, at least just sort of the basic food web, doesn't capture is the strength of interaction. So here I have a figure about what tern chicks, and terns are basically seagulls, what tern chicks eat. And you can see that they eat insects and fish and crab and lizards and other chicks and something that's not other chicks, some other stuff. But they don't sort of eat the same amount of everything. So if I have a tern up here, I could draw a food web where it eats crabs and it also eats lizards. But the truth is it's eating a ton more crab than it is lizard. And a basic food web doesn't tell you that. It's just going to show you that crabs and lizards are both eaten by terns, and you have no idea that one's eaten more or less. You have no idea that strength of interaction between those two trophic levels. Another problem with food webs is that they don't account for species that span two trophic levels. So something like a hemiparasitic plant. We learned about this in a previous lecture. These guys can photosynthesis, so in some ways they're an autotroph, but they can't sort of get everything they need just through photosynthesis. They actually sort of steal some of the carbohydrates that they need from other plants. And so are they a primary producer? Are they a primary consumer? Where do you put them in the food web? It's a little unclear, and that's because food webs, again, are a little bit too simplistic to capture the true messiness of ecological communities. Another problem that's sort of related to food webs, or with food webs, is that you have many species that feed at multiple trophic levels. So just because I'm a hawk and I might take a snake that's a tertiary consumer doesn't mean I'm not going to take a mouse that's a secondary consumer. And just because I'm a mouse that tends to eat some grasshoppers, so that makes me a secondary consumer, doesn't mean I'm also not going to take some seeds. I might be an omnivore, and omnivores feed on both plants and animals, and so it's harder to put them into that food web, at least in a chain-like food web where you're sort of moving up from primary producers all the way through to catenary consumers. Another problem with food webs is that some important food resources, mostly detritus, are not represented. So detritus is organic matter, dead animals, dead plants, dead stuff that died before they got eaten by something else. And so they're sort of decomposing, they're breaking down, bacteria and fungal organisms are breaking them down, and those are called detritivores. In fact, any organism, even if it's a bigger organism that eats detritus, is called a detritivore. And so as they decompose, they sort of provide energy to the things that eat them. And it's very difficult to represent those in a food web because you can see anything can die and become detritus and then can be eaten by your detritivores. So there's a picture there of a food web where there's been an effort to include detritus. But again, it's very difficult to do so in a simple food web. The final problem with food webs is that food webs only include trophic or vertical interactions. They don't include non-trophic or what we call horizontal interactions. And so, they tell you about who eats who, but they don't necessarily tell you about things like competition or mutualisms that might happen between different organisms within that community. So here I have an example where the food web tells you that the producers are being eaten by herbivores and carnivores all the way up. But then I have what we call an interaction web that includes these other interactions that we learned about: mutualisms, competition, amensalism, commensalism. All those interactions, we can draw those in an interaction web. And frequently those interactions actually occur within a trophic level, although not always. All right. So that's food webs. Another important element of communities is classifying community structure. And there's a couple of different metrics that we use to just describe the overall broad-scale structure of a community. Two of the big ones are species diversity and composition. And within that, we also have sort of finer grade definitions. One is species richness. Species richness is just a total count of the number of species in a community. So I've got two plant communities in this slide. You see community 1 and community 2. And you'll notice that if you count up the total number of different species in both communities, there are 4. So for both of these communities, species richness is 4. Now, of course, what you'll notice is that these communities are quite different. The top one has a lot of blue Christmas trees, and the bottom one only has 1. The bottom one has tons of that deciduous-looking tree, and the top one has about 4 or 5. And so you'll notice that species richness definitely can't capture everything about the community. It doesn't tell you everything about community structure, but it goes a little ways. Another thing we can think about is species evenness. Species evenness is the commonness or rareness, or sort of the distribution of individuals across the different species in the community. So the top figure is what I would call an even community. I have about the same amount of each tree species. I have 25% of the deciduous trees, 25% of the blue Christmas trees, 25% of the other weird-looking trees, and 25% of the green Christmas trees. Whereas the bottom community, community number 2, is what I would call an uneven community. I have primarily deciduous trees with 1 or 2 Christmas trees and that one weird thing thrown in. And you can actually see evenness if you plot a bar graph. That first figure, you'll notice that if I plot the number of each individual tree, that bar graph is sort of flat right along the top. It's very even. But if I look at the community number 2, I've got way more deciduous trees, and so I have that very uneven sort of graph in that bottom figure for that community number 2. Species diversity itself, which is one of the more common metrics, that actually is a combination of species richness and species evenness. And so there's very different ways of measuring or different metrics that you can use for diversity. We're just going to look at one in this class, and that's called the Shannon Index. Regardless of the metric you use, typical metrics of species diversity can include both richness and evenness within them. They kind of ball it all up into a single number. So the Shannon Index is probably the most common species diversity index, and it's calculated according to this equation. And the H is actually the index value. Those P's are the proportion of individuals that are from species I, and S is the total number of species in the community. And I know this probably doesn't mean much to you, so we're going to go through some examples. So if I take my tree example here, my community number 1, and I want to calculate the Shannon Index for this particular community, I notice that there are 25% of the deciduous trees. So my proportion of deciduous trees is 0.25. I've got 25% that weird looking tall light green tree, so my proportion of tree number 2 is 0.25. I have 25% blue Christmas trees, so again proportion number 3 is 0.25. And I have 25% green Christmas trees, so proportion number 4 is 0.25. And if I have 4 different species in this community, I should have 4 terms in my summation for species diversity. So all I do is I multiply the proportion of the first species times the logarithm of that proportion, and then I add it to the proportion of the second species times the logarithm of that proportion, and so on and so on and so on until I've added up all of my species. And then what I don't forget the negative sign. If you get a negative Shannon Index, you've done something wrong. So I put that negative sign in, and I get my Shannon Index is 1.3863. Now I realize that means absolutely nothing to you. If you use Shannon indices for a while, it might have some meaning, but usually the most typical way that Shannon indexes can be helpful is comparing 2 different communities to try and understand whether their combined richness and evenness is really different or not. So let's take a look at community number 2. Sorry, that should say community number 2. In this case, I have 80% of my trees as deciduous, so the proportion of my first species is 0.8. And then I only have 5% of my trees being those, that tall, weird, light green tree, just one individual, 1 out of 20. And then I have another 5% is the blue Christmas tree, and 10% is the green Christmas tree. So I've got different proportions because this is a different community. Same kind of thing, though, to calculate the Shannon Index. I take the proportion of my first species, multiply it by the logarithm of that proportion. I add it to the proportion of the second species times the logarithm of the second species. I add it to the proportion of the third species times the logarithm of the third species, and I add that to the proportion of my fourth species times the logarithm of my fourth species. And again, you should have exactly the same number of terms as you have species in that community, so in this case 4. And then I don't forget the negative sign, and I get a number. And this time my number, instead of being 1.3, is 0.7, much smaller. And so the Shannon Index is smaller when the community is less even. Remember, this community number 2 was much less even. If I looked at the bar chart, it was not flat along the top. I had tons of the deciduous tree and not much of anything else. So you get smaller Shannon indices when you have less even communities. All right, let's look at another example. Here I have birds, and I have 2 different communities, community number 1 on top and community number 2 on the bottom. Now these 2 communities are actually similar in evenness. I have the same number of each species in each community. So I have 25% sparrows, 25% crows, 25% pigeons, and 25% cardinals in my first community, same exact amount of sparrows, cardinals, crows, and pigeons. In community number 2, I again have the same amount of the different birds. I have 33% pigeons, 33% sparrows, and 33% crows. So it's again completely even. The difference is I have more species in community number 1, I've got cardinals, than I do in community number 2. So let's take a look. If I look at community number 1, my proportions are all 0.25. So I put that into my equation for the Shannon index, and I get 1.3863. That's what I had before. Let's take a look at community number 2. And now I have proportions 0.33, 0.33, and 0.33. I again plug that into my equation for the Shannon index. And this time I only have 3 terms because I only have 3 species in my community. And I get 1.0976. So 1.09 is smaller than 1.3. And so my Shannon index is smaller when the community has fewer species. So your Shannon index is going to be really small when you have only a few species and you have a really uneven distribution. So mostly one thing and then maybe a couple other things thrown in. Your Shannon index is going to be really big when you have many different species and about even distribution across all of them, when most of them are about as abundant as the others. And so that's sort of what the Shannon index means. The absolute minimum Shannon index is 0. If you get a Shannon index that is below 0, you've probably forgotten the negative sign. You get a Shannon index of 0 when there's only one species in your community. So here I have 100% crows. So my proportion is 1, and that if I plug that in, the logarithm of 1 is 0. And so again, the minimum Shannon index is 0, and that's what you get when there's only one species present. And it's almost impossible to define what evenness means when there is only one species. So your richness is 1, your evenness is kind of undefined, and that gives you a Shannon index of 0. All right, so now we're going to do a worked out example in class, and I would recommend that you try to make sure, if you are not in class, that you know how to do this example. All right, so far we've talked about species diversity, and that, like we said, reflects, or at least should reflect, both species evenness and species richness. Sometimes people use the word species diversity when they actually mean richness, when they actually mean the number of species. But I want you to always remember that species diversity is a combination, a balled up metric that includes both the number of species there, but also how evenly they're distributed. Species diversity, though, is a little bit different from biodiversity. Biodiversity is not just species diversity. Species diversity is one element of biodiversity. Biodiversity, though, includes other ecological entities. So I might have a population that's all the same species, but there might be a lot of different genes in there. And so the species diversity is very low, but I have a lot of genetic diversity. And because of that genetic diversity, I would say there's still some biodiversity in that system. And so at the smaller scale with genes, we can have high biodiversity, and that's separate from species diversity. Biodiversity can also capture a higher level where I'm looking at the number of different types of communities in a region. I might have some regions where I have one type of community. It might be species diverse, but I might just have one type of community everywhere. And then I might have other regions where I have a whole pile of different communities with different species living in them scattered across the landscape. So again, biodiversity is a broader net than species diversity. All right. With species diversity, like I said, the main way we can characterize it is with that Shannon index, but that's a number. And unless you become really familiar with Shannon indices and use them in a lot of different systems, they don't have an intrinsic meaning to you. If I tell you the Shannon index is 1.3, you might not know what that means. So frequently what we do to try and understand species diversity a little bit better is plot what we call rank abundance curves. So to do this, we plot the proportional abundance of each species relative to the others in rank order from the most abundant to the least abundant. That probably means nothing to you, so I'm going to give an example. Here I have a bird community, and I have 8 crows, 6 sparrows, 4 pigeons, and 2 cardinals. So if I'm going to rank these, that just means sort of rate them in order of the one that's most abundant to least abundant. I would say my crows rank as 1 because there's most of them. Then my sparrows, then my pigeons, and then my cardinals are my highest rank because there's only 2 of them, so they have a rank of 4. To calculate the proportion, I'm just going to count the number of that species and divide by the total number of birds. So I have 8 crows and I have 20 birds, so my proportion is 0.4, 40%. I have 6 sparrows and 20 birds, so my proportion is 0.3 or 30%, and so on and so on and so on. So then what I do is I plot the rank along my x against the proportion along my y, and I can do that as a bar graph. That's a rank abundance curve, but I can also just do it as a line. That's also a rank abundance curve. And of course, from that rank abundance curve, I can actually use it to calculate my Shannon diversity index. All I have to do is I pull off the proportion along the y-axis for each different species, and I substitute it into my equation for the Shannon index. You can try that. You should get the number 1.2799 for this particular community. Let's do another example. Here's a different bird community with slightly different amounts of crows, sparrows, pigeons, and cardinals. In this case, I have 10 sparrows, 7 crows, 3 cardinals, and 1 pigeon. So if I'm ranking them, the most abundant is sparrows, that gets a 1. The least abundant is pigeons, that gets a 4. And then I calculate the proportions. I have 10 sparrows out of 20 birds, so that's 50% sparrows, or my proportion is 0.5. 7 crows gives me a proportion of 0.3. 3 cardinals, a proportion of 0.15, and 1 pigeon, that's 1 out of 20, is 0.05, or 5% pigeons. Again, I can plot this as a bar graph or a line graph. And again, I can calculate my Shannon diversity index either from the numbers that are in that proportion column or by reading it off the graph. That gives me 1.1484. So again, this community has a different species diversity than the previous one that we looked at. All right, so that's rank abundance curves. And I want to make sure that you keep rank abundance curves differentiated from species accumulation curves. Rank abundance curves are about what's there. We already have the system completely characterized. We know how many of this and that we've got, and so we plot this rank abundance curve knowing everything about that particular community. Species accumulation curves are different. They are graphical representations of how the detected species richness changes with sampling effort. And the idea here is that the more you go out and sample, the more you're going to find different species. So if I go to downtown Clemson today and I bird watch, I'm going to have some number of species. If I go tomorrow, I might find some new species I didn't see today. And if I go out the next day, I'm going to find some new ones I didn't see on the first 2 days, and so on and so on and so on, until you've sort of seen all the birds that you're ever going to see here, and your curve starts to flatten out. So when you first go out and do some sampling in a new area, you find a lot of new individuals, a lot of new species you've never seen before. But as you do it more and more in the same area, there's diminishing returns because eventually you've just seen everything that can possibly live there that you can possibly find. And so we represent that with these species accumulation curves where we plot sampling effort or how many times you've gone out and bird watched or how many years you've been doing a monitoring project as a function of, you plot that along the x and you plot the number of species you found, that's richness, just the total species count, along the y-axis. So curves from different environments look different, and we sort of classify them into 2 groups. We call saturated curves, ones where the number of species you're getting per sampling time, per sampling effort, is kind of flattened out. So, you've got a lot of species initially at first, and then now that you keep going out and searching for more species, you're not finding much new. And so eventually the curve flattens completely, and you found every species that's there. An unsaturated species accumulation curve, though, is more like that straight line up the middle of the graph for tropical soil bacteria. Here I go out today and I find 10 new species, and I go out tomorrow and I find another 10 species, and I go out the next day and I find 10 new species again, and I keep going out and finding new species I've never seen before. And so in that case, eventually, if you go out and sample enough times, the curve should turn over, but until it flattens out, we call it an unsaturated curve. And that's an indication that there's still a lot of species in that community that you don't know about. All right, so we can do that with individual communities. We can also do this sort of accumulation curve across the whole entire globe. And there's a little bit of shadiness with this kind of approach because we obviously have not sampled the entire globe. We're obviously still in that quite unsaturated part of the curve where every time we go look we find new stuff, but because we maybe started to turn over, what scientists have done is extrapolated and tried to fit the curve to figure out where exactly we're going to flatten out and how many species there are on Earth. And so when I said we really defined 2 million species, but there's 9 million on Earth, this is how scientists get that number of 9 million. It's extrapolating these curves and saying, well, it looks like, with enough sampling effort, we're going to saturate at about 9 million species. So that must be how many there are on Earth. All right. So that's a bit about species diversity. But another element of a community is species composition. If I take a look at those two communities, community one with the plants and community one with the birds, both of these communities have the same richness. They both have 4 species. They actually have the same evenness because there's 25% of each of the 4 species in both communities. But obviously, just looking at these two communities, the bird community and the tree community, I know they're different. And they're different not because of one of these diversity metrics, but because of the fact that they're actually just different species involved. And so for that, to try and describe which actual species are in the community, we use the term species composition. That's the identity of the species present in the community. And you don't get that out of richness metrics or diversity metrics. But it is important for understanding the community and understanding how the community functions. If I want to know what that community is going to do, how it's going to respond to climate change, how it's going to respond to putting a road through it, I really need to know about those individual species and what they're doing and how they live. So again, that's species composition. All right, so we have species composition, species diversity. These are typical ways we characterize species. And then once we've done that, we start digging down into the community and looking at the interactions among the species populations within that community. And we've already seen this figure where we show that species interactions have a huge effect on what the community looks like. It has a huge effect on your how many of each species you find in that community, on the evenness of the community, on the species richness of the community. And this example is with the wolves in Yellowstone, where a few wolves were put into Yellowstone, and the wolf population went up, and that had a direct effect on the elks. The wolf ate the elks, and the elk population went down. But then there were all these other indirect effects where because there were less elk, there was less grazing on aspen, there was less grazing on cottonwood, there was less grazing on willow. So those populations went up. And then when those plant populations went up, the beaver population went up. And in fact, also the bison population went up because there wasn't the competition with elk and there were more plants than the bison could eat. All right. So that brings us to this dichotomy in terms of species interactions, where we can think about direct species interactions and indirect interactions. And in the previous unit, we focused on pairwise interactions between species, and those were all mostly direct. We were interested in one species predating on another, or one species directly competing with another, or one species directly helping another. But now that we're thinking about these broader, larger communities where we have all sorts of interactions going on, we need to think about direct versus indirect. Indirect species interactions occur between 2 species and include both trophic, both feeding, and non-trophic, non-feeding interactions directly between one species and the other. So here I have my wolves. They interact directly with elk. They feed on elk. They sort of actually munch into the elk body. And so that is a direct interaction. It's a direct trophic interaction. An indirect interaction occurs when the relationship between 2 species is mediated by a third or more species. So the 2 species that have an interaction may never even meet. They may never see each other. They may forage at different times of the year or different times of the day or in different places. But because of these other species that are kind of linking the whole network together, those 2 species actually do matter to one another through a third or other species. And here we have the example. We have the wolves that have a direct trophic interaction on the elk. We have the elk that have a direct trophic interaction on cottonwood. And so indirectly, wolves affect the population of cottonwood. The wolf never eats cottonwood. The wolf doesn't even necessarily come close to the cottonwood, but it has that effect on cottonwood through the elk. So again, that's the interaction between wolves and cottonwood mediated by a third party being the elk. Although indirect interactions may seem kind of loose and weak, they're actually not. They can be very important for determining community composition, as we see in this Yellowstone example. They can actually alter how species interact. They can turn negative interactions, like predation, into positive interactions, like removing a competitor, and that sort of thing. So indirect interactions can be very, very important in determining what a community looks like. And they can also be very difficult to study and think your way through, well, how's that going to affect this, and how's that going to affect that? And that's what makes community ecology so interesting and exciting, is that it's just so complex, all these indirect ripple effects, basically, that you can have in a community when you change one population. So that leaves the idea of trophic cascades. This is one type of ripple effect. And a trophic cascade occurs when the rate of consumption at one trophic level results in changes in species abundance or composition at lower trophic levels. So again, going back to the Yellowstone example, we have the wolves feeding on the elk, that's one trophic level, but then we have this trickle-down effect to the cottonwood at a second trophic level further down that indirectly results from the wolves. And so that's a trophic cascade. It's going from not just between two trophic levels that are directly adjacent, it's going all the way down to the bottom primary producer level of the cottonwoods. So again, there's our secondary consumer, that's the one level. The primary consumer, that's just our direct interaction, and then we have our trophic cascade down to the primary producer. Another type of interesting interaction you can get in communities with trophic effects is what we call trophic facilitation. So this occurs when a consumer is indirectly helped by a positive interaction between its prey and some other species. And here's an example where I have aphids, and I have two different fruit, juncus and iva. So aphids eat iva. Aphids do not eat juncus. But juncus is still beneficial to aphids because it actually helps iva to grow. So juncus shades the soil and prevents evaporation, which then prevents salt buildup, and this is helpful for iva growth. Juncus also oxygenates the soil, and again, this is helpful for iva growth. And because the juncus helps the iva to grow better, that actually helps the aphids to do better. So even though aphids don't eat juncus, they still do better with the juncus present, and that's called trophic facilitation. So again, aphids on iva grow better when juncus is present, even though they don't eat it. So just like we can have these messy direct and indirect interactions in trophic networks, we can also have those in other types of interaction networks, something like a competitive network. So here we have horizontal interactions, not vertical, not feeding interactions, not trophic interactions, and typically these do occur at a single trophic level, like I said before, but they're really important, even though they're not feeding interactions. They're really important, and one of the reasons they are is that they often are used to explain species richness. So frequently we think of competition and competition networks as being the determinant of how many species can coexist together. We talked about coexistence in pairwise interactions, but then as we get to bigger and bigger communities with more and more species, you have to think about how all those different species manage to live in the same place at the same time without outcompeting each other. And one of the solutions for explaining this that scientists have come up with is the idea of balance. And it's basically like a rock, paper, scissors game. So in the one case, you might have a competitive hierarchy where A is better than B and B is better than C. That doesn't necessarily lead to coexistence as easily as when you have a competitive network where A inhibits B, B inhibits C, but then C turns around and inhibits A. So again, it's like rock, paper, scissors. Scissors is never the best, rock is never the best, and paper is never the best. You never know what's going to be the best because it's that tripartite interaction between all three. And again, that's common, it's a common motif in competitive networks, and scientists believe that it's probably an explanation for why you can have multiple different species living together. Some people have tested that the hypothesis that these sort of triangles actually do lead to coexistence and higher species diversity, and they actually found that these little tripartite interactions where each species inhibits the other in circles are quite common in some of these diverse systems. All right. So whether we're thinking about trophic interactions, competitive interactions, even mutualistic interactions, you can have mutualistic networks. I didn't show any, but you can have that. Regardless of what type of interaction we're talking about, one important metric is interaction strength, which is the effect of one species on the abundance of another. And the easy way to measure interaction strength between two species is to use what we call an exclusion experiment. We talked about that a little bit in a previous lecture. Basically, we remove one species, the interactor species, and we look at the effect on the other species, the target species. And the interaction strength varies widely among species. So sometimes the interaction strength is 0 and those two species don't interact at all. Sometimes it's a really strong interaction and the one species really helps or really hurts the population of the other species. And sometimes it's a weak interaction. Yes, there's less of the species B because species A is present, but not that much less. So again, we think about that turn, feeding on crabs and feeding on lizards, And we think, well, the interaction strength between terns and crabs is much stronger because they eat a lot more crabs than the interaction strength between terns and lizards. So to actually put that in quantitative terms, we can calculate what we call the per capita interaction strength. And your textbook has a terrible, terrible, terrible version of this equation. I warn you not to really look at the textbook. It's hard to tell what's supposed to be in brackets with the textbook. So this is the actual equation. We basically take the logarithm of C over E and divide it by I. So C is the number of target individuals, the ones that are going to be affected by this other thing, that are in the presence of the interactor. E is the number of target individuals in the absence of the interactor. So again, the number that are going to be affected by the other guy. And I is the number that are doing the affecting, the number of interactor individuals. So again, I know that's a little bit hard to understand, but in class we did work through an example, so I would encourage you to get class notes or to make sure you know what you're doing in terms of calculating interaction strength, in terms of using that equation, which numbers go where in that equation. All right, so the final thing we're going to talk about with this introduction to communities is the different types of species that are very important to communities and community structure, how the community looks. The first is a foundation species. So this is a large or abundant species that has a large community-wide effect by providing habitat or food. And we talked about beech forests previously. Beech is a good example of a foundation species. It provides habitat and food for the animals that live in the beech forest. And it sort of defines what that whole forest is, what that whole community is made up of. It's a little different from an ecosystem engineer. An ecosystem engineer is not necessarily large or abundant, but it's really good at creating, modifying, or maintaining the physical habitat for themselves. And so, not just because I'm Canadian, one of the most famous ecosystem engineers is, of course, the beaver. Because the beaver just decides to make a lake here, or a pond here, or redirect that river over there. And so it's really changing and modifying that whole physical environment to meet its own needs, but in doing so, it's sort of changing it for all the other animals that live in that community, all the other plants and animals that live in that community. The final type of species is a keystone species. So these are species that have large effects, not because they're abundant, not because they're necessarily having drastic effects on the physical habitat, but because they play vital roles in the community. So one example is the wolf in Yellowstone. We introduced, I think, maybe 10 wolves to start, and within a couple of years we had a dramatic effect. There weren't tons of wolves, they weren't really abundant, they're not super large, they're not really altering the environment that much, but what they are doing is they're playing that role as the top predator, and that's really affecting all the trophic levels below them, and so that's what we would call a keystone species. All right, so I hope you've enjoyed our introduction to community ecology. If you have any questions, I'd encourage you to email me. Otherwise, I will see you at the next lecture.